Hello.
Hello. No, hello Hiroshi, hello Shinya. Now that you start the broadcast, everyone can hear what we're saying. So that's fine.
Yes, I did. Was it a problem?
No, if you don't start the broadcast at the first place, you can first discuss and then start the broadcast, and everyone who joins can automatically hear you.
Okay.
But it's fine. Well, welcome. Good morning. Or good afternoon.
Yeah, good morning.
Good evening, probably, rather.
No, not yet. It's 3:30. So.
Very good.
Very
good. Are you doing fine? Everything okay? Anything you need?
I'm fine.
You're very good. Hiroshi, you too?
Yes, everything's fine.
Very good.
Except that I did some rehearsal yesterday and found that I had too many slides. So I need to cut 2 slides explaining the rescue function.
Yeah, that's fine. That's fine.
I
In
hope
the
it's
end,
fine.
Yeah, sure. As long as people, it's fine. It's fine. It's fine. It's good. No worries. No worries. No worries.
Okay, hear my voice?
Yes, we can hear your voice. Yes.
Okay.
So we have in total now 30 registered attendees.
Yeah.
That will be, yeah. Good. Now we're doing here. Start this.
"30 people registered. So we have 30 audience."
Yes. At the moment, yes. Yes.
Is it possible to join without registration?
Yes. You can just, if you as soon as you have the login. So if you have someone in your lab, or if you have someone you say it might be interested, just send them the link. It's this webinar ID on the lower right side. You can also click. Very good. Now I actually see you. Can you see me? No, you can't. Now you should be able to see me. Hello.
I do. Hi. Yes, now I see.
Very good. Sorry about that. Thank you again, Hiroshi, for doing this. Thank you for taking part.
No, my pleasure. My pleasure. I hope I will attract people.
For sure.
At least I did my best.
I'm pretty sure of that.
Marty, I have one question.
Yes, please.
So can I use my own background on the GoToWebinar?
I think not. Yeah, that's an issue.
Okay.
But I think your background is very pretty, so you could, in this case it should be an issue. But yes, you can't blur it. You mean make it fuzzy? Yeah. I miss that feature as well. I miss it as well.
Okay. So firstly, I talk in Japanese a minute, and then after that I will explain shortly in English.
Oh, someone joined and immediately left again. But yes, that's good. Sounds good. Very good. So we have half an hour left. Oh, yeah, we do.
Yes, that's right. Can
Would you like to
quickly go through?
Yes, of course. Can you already share your screen, or do I need to give you permission?
Very good. Do this.
Please do so.
All right.
I need to quickly connect you with my laptop.
How can I do this?
To share your screen, or
Yes. Yes.
it should be only in the upper right corner. I make you presenter. Maybe that helps.
Yeah, I find something. Yes, now I'm the presenter.
Yes.
Then I will display my screen.
Yes.
Then my display shown now.
Yes.
Right?
Yes. Feel free. Please try. Very good. I see the Gasthof and I see the abberior. Yes.
Yes, I think it's Regensburg that it shows. All right.
Very good. Very good. Glad that you like them. Chrome.
Is everything fine on your display?
Yes, looks very good.
Yes.
Yeah, some of the movies still slow.
Yeah, but it's fine. But no, the speed is actually fine. Very good. Shinya?
Yeah. Yes, I can hear you.
If we turn off our webcams, the image gets a bit bigger for the PowerPoint presentation.
Yes.
Yeah. So in case, or tell people, please, if you can at the beginning, they can also hide the webcam. So stop sharing the webcam there. So what they see, so they have a bigger screen. Yeah, because with microscopy images, you want it as big as possible. And I just checked it on the microscope on my laptop, and of course we are visible on the top bar of the screen, and we take up a bit of space. So it might be helpful for some people to maximize the slides and minimize us.
花村さん、カンタムデザインってこれ間になんか入れませんよね。
あ、点が入りますけど大丈夫ですよ。
あ、マジですか。ちょっと直します。Sorry, Martin. I just asking him.
Please feel free. I have no.
The name of the quantum design. So I found that I need dot like this.
Yep. Looks good.
Then
but please speak as much Japanese as you like. Really, I don't feel excluded at all. Really, it's I expect you to.
But I think it's a bit weird to.
No, it's fine. That's fine. Really.
Fine.
It's fine. I trust you, so I know you do not talk strange stuff. So yes, Shinya, please.
Martin, yeah. So I want the audience to focus on the slide. So I think it's better to turn off our web camera.
Yes. The thing is, it's a question whether Hiroshi's webcam should also be turned off because he's the speaker, or not. I will turn my off. Yes.
Okay, 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 okay. Understood. Okay, okay.
But I agree. I agree with you.
Should I turn off mine?
No, it's
turn
a,
off
it's a matter of debate. You can, but you don't need to. You are the speaker. Usually people want to see you.
Yes.
Of course, with the mask, it's kind of
No,
actually.
I will get rid of this. I will
Yeah.
not use this.
No worries. No worries. It's
Ayumi, your name is Akira Shiraishi.
So just for you, for interest, I have it on my laptop open, and people can see and hear everything we say now. Yeah. So as soon as someone joins, yeah, they will be able to hear everything. Yeah, it's an open discussion. Just that you know that if someone joins, we need to maybe. Normally, it's better to start the webinar only 5 minutes beforehand. Yeah, so it's easier that we can discuss and then let people in 5 minutes before. But it's completely fine. We'll just leave it like this, and everything will be good. Very good. So, Hiroshi, I would quickly share your screen and make me presenter. Yeah, so I can.
Yes.
I can show PowerPoint like so. I see everything here. Yes, it's good.
Oh, Martin, I'm not going to use the image that you sent me. Sorry.
No worries. It's just an offer. No, it's fine. It's fine. No worries. It's just if you want to use it, feel free, but absolutely no issue. You do your slides as you like them. That's the thing, and you feel comfortable with.
Thanks.
So webinar this one is working, and now the PowerPoint, this one. Especially here for the microscope software, it's useful if we then at some point switch off all our cameras, because then it's easier to actually follow. It's a little bit lagging, but it will be okay. So let me check.
Wow.
Let's see whether that's good.
花村さん。
はい。
僕のプレゼンが終わった後なんですけど、
はい。
ウェビナー終わるまで僕ログインしてた方がいいです？
いや、大丈夫ですよ。
離れてもいいです？
はい、大丈夫ですよ。もし、なので、川辺先生のセッションが終わって、一応質問コーナーを設けて、で、それが終われば、もし、何か川辺先生に対してというのがありましたら、また私の方から連絡させていただきますので。
そうですね、直接メールを送っていただいても結構ですし。
了解いたしました。
30人がもっと集まるといいですね。
そうですね。結構、いろいろ声をかけたんですけど、あと結構オンディマンドで配信してほしいっていう先生方も結構案内したらいらっしゃったんで、
そうですか。
確か
にこの4時ってね、ちょっと大学だとちょうど会議がある時間帯でもあるんですよね。
あとやっぱ結構臨床系の先生とかもすごく興味を持ってくださってる先生が結構東大とかでもいらっしゃって、オンディマンドで改めて配信してほしいっていう結構リクエストとかはいただいてたんで、またここからまた拡散していくとは思います。
素晴らしい。
So we already have 2 guests who can actually hear us. So if you want at some point introduce us, that's also fine, but we can also wait until the webinar starts officially in 20 minutes.
We already have 2 guests. It's 20 minutes before the starting time. It's great. Right. まだ時間ありますね。ちょっとトイレ行ってきます。
はい。
Martin?
Yes, please. Yes.
Can I ask the technical question or
Yes,
shall you
please.
do it later?
No, feel free.
I'm looking for an antibody against guinea pig and also rat.
From abberior or secondaries?
No, I'm sorry, but it's from other company. So
yeah, sure.
I prefer Alexa 594 for the red
Yes.
channel.
Yes.
I will send an email, but could you later recommend me which antibody is the best in your hand?
For the So of course there's abberior star orange, but do you mean what would be the best for the red range general, or?
No, the which antibody conjugated with 594 is the best. The problem is that I need to get the product in like 5 days from some technical reasons.
Yeah,
So
sure,
if
sure,
I
sure.
order the antibody from a company and it may not be in time, so I need to order the one from the, the
Yeah, sure, sure.
with
So
Alexa
594. Alexa
Yes.
594 is good. Alexa, wait, let me something.
So I just wrote an email and I'm sending you.
Yeah, please
I
send
just
it
want to
and
know
then
after the thing right now.
yes.
Mm-hmm.
I think 594 would be one of the absolutely preferred choices. Yes, 594 is good. We often use 594 as well.
Yes. So if you have the information of the catalog number of these antibodies, please, there are many products from many different companies.
Yeah, sure.
With Alexa 594.
Well,
It's really confusing.
What is your typical supplier? Where would you buy it usually?
Usually I buy from some official, I think. But later, after this webinar.
Yes.
Now let's focus on this. Oh yeah. Mhm. Mhm.
それでは、時間になりましたので、始めさせていただきます。本日ですね、アッベリアインストゥルメント社と、日本カンタムデザインによります、超解像顕微鏡ウェビナーを始めさせていただきます。本日は、多数の方にご参加いただいております。ありがとうございます。本日は、このステッド顕微鏡のウェビナー開催させていただきます。日本カンタムデザイン、私、アッベリア超解像顕微鏡の販売担当、花村と申します。カンタムデザイン社は、アッベリアインストゥルメント社の取り扱う超解像顕微鏡のデモ、販売、納入からアフターサポートまで行っております。本日はアッベリアインストゥルメント社と弊社日本カンタムデザイン社によります合計3回のウェビナー、その第1回目となります。本日は神経科学者のためのアダプティブオプティクスステッドと題しまして、アッベリア製のステッド顕微鏡のユーザーでもいらっしゃいます、群馬大学医学部薬理学教授、川辺博先生より、ステッド顕微鏡の原理、アプリケーションを日本語にて紹介いただきます。その後、アッベリアインストゥルメント社のマーティン・メシュカットによります装置紹介、オンラインライブデモ、こちらを英語にて紹介させていただきます。質問は英語、日本語、どちらでも構いませんので、質問ボックスにご記載いただいたら結構です。My name is Shinya Hanamure from Quantum Design Japan. Quantum Design is a distributor of abberior instruments in Japan. Today, abberior STED user Professor Hiroshi Kawabe from Gunma University will explain the principle of STED microscopy and introduce the application in Japanese. After that, Martin Meschkat from abberior instruments will introduce the latest function and the online lab demo in English. Questions are accepted in both English and Japanese. それでは川辺先生、どうぞよろしくお願いいたします。
はい、よろしくお願いします。画面私の見えてますでしょうか。見えてますね。これで大丈夫ですよね。はい。それでは始めたいと思います。花村さん、ご紹介ありがとうございます。群馬大学の川辺です。本日は神経科学研究での 3D ステッド顕微鏡の応用ということでお話しさせていただきます。人の脳内神経ネットワークはこの図にあるように非常に複雑です。 神経ネットワークが記憶や学習などの工事機能を担いますが、その際シナプスを介して情報のやり取りが行われます。シナプスを介した情報伝達の異常が自閉症スペクトラム障害、統合失調症、あるいは双極性障害の発症につながると考えられており、シナプス形成の制御は非常に重要な研究分野です。シナプス形成に至るまで5つの神経細胞発達過程があります。まず最初に再生中期に神経管が閉鎖しますと、神経管の上皮細胞である、そして神経管細胞、ステムセルであるこのラディアルグリア細胞が非対称分裂によってこのように新生神経細胞を生み出します。この過程が一番最初の神経新生の過程です。生まれたばかりの神経細胞というのはこのように、同じような長さの突起が四方八方に突き出ておりますが、2番目の極性形成のステップで、この神経細胞がラディアルグリア細胞に付着することで、先導突起と、そしてトレーニングプロセス、こういった2つの突起を持つ細胞へと形を変えます。そして3番目のステップが、脳の深い位置から表面に向かって、ラディアルグリア細胞に沿って神経細胞が移動する過程です。目的地まで到着しますと、この神経細胞がラディアルグリア細胞から離れまして、そして樹状突起と軸索を伸ばしていきます。この過程が神経突起成長の過程です。そして最終的に軸索と樹状突起がこのように接触するところにシナプスが形成されます。この部分を拡大したのがこの右の図になります。軸索終末には神経伝達物質が詰まったシナプス小胞があり、そしてシナプスの樹状突起側、すなわちスパインの表面には、その細胞膜に神経伝達物質の受容体が濃縮しています。そして、その細胞質はシナプス高飛行 PSE によって裏打ちされております。これら5つの発達過程の制御に重要な細胞骨格とシナプスの構成タンパク質の極材を明らかにしたいというときに一般的に行われるのが免疫細胞染色、免疫組織染色です。こちら神経細胞の模式図ですが、こちらを拡大したのがこちらです。細胞外と細胞内が細胞膜、脂質二重膜によって隔てられています。まずパラフォルムアルデヒドでタンパク質をこのように加強してやった後に、界面活性剤で細胞膜を溶出します。そして特定のタンパク質、この場合このドラム缶が半分になったような形のタンパク質ですが、これに対する抗体を変化する。そうすると抗原抗体反応で複合体ができるわけです。次にこの抗体を認識する蛍光標識した二次抗体を変化すると、間接的に特定のタンパク質が蛍光標識できるということになります。この蛍光の極材位置を観察することで、この特定のタンパク質が細胞内のどこにあるのか、これを観察するのが蛍光顕微鏡です。蛍光顕微鏡には分解能というパラメータがあります。左側の図、この左側に2つの蛍光の光源があるとします。これ、カーソル見えてますかね？
見えてます。
大丈夫ですか？
はい、見えてます。
蛍光の光源がこちら2つあります。で、この光源から出てきた蛍光がレンズを通して、そして検出器に収蔵します。この際、蛍光は波としての、光ですので、波としての性質があるために、一点に収束するということはありません。ある程度の広がりを持って、このようなピークを描きます。で、この蛍光の光源、この2つの距離が十分離れている場合、検出器に検出されるこの2つのピークは2点として分離することができます。しかし、この蛍光光源の距離が短くなっていきますと、だんだんこのピークの間の距離も短くなってきて、最終的には分解できなくなります。2つの光源を分解できるギリギリの蛍光光源の距離、これが分解能です。分解能にはいくつかの定義があるんですが、その一つとして代表的なものとして 0.61× 光の波長、これを開口数で割ったものがあります。この開口数というのはレンズの大きさに依存している値です。 つまり蛍光顕微鏡の分解能というのは、光の波長とレンズの特性のみに依存しているということが言えます。これは100年間以上信じられてきた顕微鏡の限界です。この限界を打ち破ったのがこの3人の先生方です。この先生方は超解像蛍光顕微鏡を開発した功績で2014年にノーベル賞を受賞されました。私はこの中でもステファン・ヘル先生が開発した超解像 3D ステッド顕微鏡を研究に用いております。この顕微鏡の原理を説明する前に強焦点顕微鏡、一般に研究室でよく使われている強焦点顕微鏡について説明いたします。まず蛍光物質が規定状態にある蛍光物質に冷気レーザーが当たりますと、この蛍光物質、冷気状態に上がります。そしてエネルギー陣を少し落とした後に規定状態まで戻るときに蛍光を発生します。この B の図は強焦点顕微鏡の航路図です。冷気レーザーが大距離区内に当たって反射して、そして対物レンズを通して標本に当たります。そしてここから出てきた蛍光が大距離区内を通過して検出器に検出されるということになります。この際、冷気レーザーはこれもまた光ですので波としての性質があります。ですので、どんなに性能のいい対物レンズを使ったとしても、ある程度の広がりがここでできてしまいます。このような広がりを持ったすべての領域、ここから蛍光が出てくる可能性があるわけです。この広がりのことを点分布関数と呼んでいます。ステッド顕微鏡のステッドとは、Stimulated Emission Depletion の略で、日本語では誘導放出と言われています。冷気状態にある蛍光物質、ここに超波長の光が当たりますと、この蛍光物質が強制的に規定状態まで押し戻されます。ここでポイントなのは、この誘導放出の際に蛍光は発生しないということです。B の図にありますように、ステッド顕微鏡というのは非常に単純な構造でして、強焦点顕微鏡に1本だけレーザーラインが加わっただけです。それがこのステッドレーザーです。このステッドレーザーが位相マスクを通過して大黒組合に反射し、そして冷気レーザーと同じラインに乗って標本に照射されます。ここでシュテファン・ヘル先生が工夫されたのはステッドレーザーの形です。ステッドレーザーはこのようにドーナツ状のリング状の形をしています。冷気レーザーとステッドレーザーが同時に蛍光色素に当てられますと、この中心部、ここは冷気レーザーのみが当たっていますので、蛍光が発生します。一方、この周辺部、こちらは冷気レーザーとステッドレーザーが同時に両方当たっていますので、誘導放出が起きます。そして蛍光が発生しません。そのため、点分布関数がこのように小さくなります。では、なぜこれで解像度が上がるのかということをこちらのアニメーションで説明したいと思います。ここでは2つの蛍光物質がスライドガラスの上に2つ並んでいると考えてください。まず、この2つの蛍光物質を強焦点顕微鏡で観察します。そうしますと、1つ目の蛍光物質、そして2つ目の蛍光物質から同時に蛍光が出て、検出器の上にはこのようなブロードなピークが観察できます。次に、ここにステッドレーザーが加わりますと、この中心からだけ蛍光が出ますので、1つ目の蛍光物質、2つ目の蛍光物質からのシグナルが検出器には2つのピークとして観察できる。このようにして解像度、分解能が上がるというわけです。では実際にこの顕微鏡を使ってどういうことができるのか、いくつかの例を紹介させていただきます。まずこちらの例では海馬 CA1 領域の神経細胞を蛍光色素によって標識したものを観察しています。この神経細胞の例えばこのような領域を強焦点顕微鏡で、つまり冷気レーザーのみを使って操作スキャンしました。その結果がこちらにあります。横に走っているのが樹状突起で、その表面に1、2、3、4、5個のスパインが観察できます。この部分を拡大したのがこちらにあります。 全く同じ領域をステッド顕微鏡で観察した結果がこちらです。つまり、レイキレーザーとステッドレーザーを使って観察しました。この図を見て一見して分かっていただけると思うんですけれども、ステッド顕微鏡を使うことによって、樹状突起とスパインのエッジですね、こちらがかなりシャープになっています。また、この四角で囲んだ部分、ここを拡大しますと、強焦点顕微鏡では1つのスパインであったように見えましたが、実際には1つ、2つと2つのスパインがあることがはっきりと分かります。さらに、こちらのスパイン、下の方のスパインですね。このヘッドがお椀状にくぼんでいることがはっきりと分かります。こちらも同じようにくぼんでいます。このように超解像ステッド顕微鏡を使うことでスパインの微細形態を観察することが可能になります。次にこちらの例では、成長中の神経細胞、軸索の先端にある成長円錐の構造を観察しました。ここにあります微小管の構造を観察しています。微小管は下の図にありますように、アルファチューブリンとベータチューブリンが組み合わさってできた管状の構造です。細胞骨格になります。今回の実験では、アルファチューブリンに対する抗体を使って免疫染色しました。左側の強焦点顕微鏡を使った画像ですでにこの繊維状の構造がなんとなく見えるんですけれども、超解像ステッド顕微鏡を使いますと、一本一本の微小管、こちらがはっきりと分離することができます。また、一つの微小管の幅を測定しますと、強焦点顕微鏡を使った場合、ここからここまでの幅が250ナノメートルであるのに対し、超解像ステッド顕微鏡を使いますと、このように80ナノメートルの幅に観察できます。電子顕微鏡を使ったデータから微小管の直径が約25ナノメートルであるということが分かっています。さらに、免疫染色に使っている抗体の大きさが1分子14ナノメートルであるということ、そしてこの顕微鏡の分解能が35ナノメートルであるということを考えますと、この80ナノメートルというのは非常にリーズナブルな値だと思います。このように超解像ステッド顕微鏡を使うことで詳細にタンパク質複合体の構造を定量することが可能になります。次の例として、軸索ですね。この軸索を F アクチン結合タンパクであるスペクトリンに関して傾向染色した結果をご紹介します。左側が強焦点顕微鏡で、右側が全く同じ視野を超解像ステッド顕微鏡で観察した例になります。強焦点顕微鏡を使いますと、なんとなくぼんやりとシグナルがべっとりと映るんですが、超解像ステッド顕微鏡を使いますと、アクチンとステッドリンが互い違いに規則正しい周期構造をとっているということがわかります。この実験自体は2013年のこの論文の追伸なんですけれども、このデータをもとに今現在考えられているモデルというのは、この軸索の筒状の脂質二重膜、細胞膜がアクチンとスペクトリンのこのリング状の構造によって裏打ちされているというふうに考えられます。このように超解像ステッド顕微鏡を使うことで、これまで未知であった構造も発見することができます。こちら神経細胞を使った染色の例ではないんですけれども、アフリカツメガエルの卵を使った免疫細胞染色の例を説明いたします。この実験では2つの角膜高タンパク質 GP210 と PAN4/5 の抗体を使った染色です。左側が強焦点顕微鏡で、右側が超解像ステッド顕微鏡で全く同じ視野を観察した結果になります。この部分がこちらに相当します。超解像ステッド顕微鏡を使うことによって、角膜孔のかなり正確な構造を観察することが可能になります。このようにこの顕微鏡は神経細胞以外の細胞にも応用可能な顕微鏡です。とここまではですね、ステッド顕微鏡のいいところをかなり強調してきたんですけれども、ステッド顕微鏡にもいくつか欠点はあります。こちらは各蛍光顕微鏡の点分布関数、解像度を比較したものです。この中心に一つの蛍光の点があったときに、検出器にどのようなシグナルとして観察できるかと、検出できるかということを表しています。まず一番左側が強焦点顕微鏡なんですが、この中心に一つの蛍光があったときに、XY 方向では、こちらにありますように、180から250ナノメートルのシグナルの広がりが出てしまいます。そして Z 軸方向では500から700ナノメートルのシグナルの広がりが出ます。一方、ステッド顕微鏡では、 こちらにありますように、XY 平面ではシグナルの広がりが35ナノメートルと、かなり分解能上がっているんですけれども、Z 軸方向では全く改善が認められず、700ナノメートルです。ステッド顕微鏡以外の超解像顕微鏡と言われている SIM、そしてパルム、ストームといった顕微鏡。これらすべての顕微鏡でシグナルが Z 軸方向に伸びて観察されます。ではなぜステッド顕微鏡でこのように Z 軸方向の解像度が悪いのか、これから説明いたします。これはすべてステッドレーザーの形に起因しています。下の2つの写真はステッドレーザーの形を表しているものですが、上のパネルにありますように XY 平面ではステッドレーザー、ステッドドーナツという構造をとっています。ところが横から見ますと XZ 平面ではドーナツというよりはむしろ筒のようなバームクーヘンのような形であるということがわかります。つまりこの Z 軸方向に解像度が上がる工夫が一切されていません。この顕微鏡で直径40ナノメートルの蛍光ビーズを観察しますと、こちら X、Z 軸ですが、表焦点顕微鏡ではこのようにラグビーボールのような形として観察できますが、ステッド顕微鏡では X 軸方向にはかなり解像度が上がっているんですが、Z 軸方向には改善は認められません。私たちが群馬大学で使っているステッド顕微鏡は 2D ステッドモードと 3D ステッドモードを切り替えることができる顕微鏡です。3D ステッドモードにしますと、XY 平面ではこのようにステッドドーナツがあるんですけれども、XZ 平面ではこの中心の上と下に非常に強いステッドレーザーのスポットがあります。この2つのスポットによって Z 軸方向にシグナルが広がるのを防いでいるということです。これで実際に先ほどの蛍光ビーズを観察しますと、この右下のパネルにあるように、X 方向の解像度、若干犠牲にしているんですけれども、Z 軸方向のシグナルの広がりが完璧に抑えられています。このように、ステッド顕微鏡の1つ目の欠点である Z 軸方向の解像度が低いという点に関しては、3D ステッド顕微鏡で解決することは可能になります。2つ目の欠点として退色が激しいという欠点があるんですが、今日はお時間の関係で説明できませんけれども、私たちが使っている顕微鏡には退色を最低限に抑えるための工夫もシステムも備わっております。これが私たちが現在使っているステッド顕微鏡です。こちらにありますように、この顕微鏡には2種類の冷気レーザーが搭載されています。つまり2チャンネルの超解像イメージングが可能な顕微鏡です。こちらの実験では、マウス脳組織で立体的に興奮性シナプスを可視化するという実験を行っています。マウスの大脳皮質の切片をシナプス全部のマーカーであるバズーン、そして興奮性シナプスのシナプス構部のマーカーであるフォーマー1で染色して表焦点顕微鏡、3D ステッド顕微鏡で観察した例です。こちら2つの顕微鏡で全く同じ視野を観察しております。表焦点顕微鏡で観察しますと、例えばこれ一つのシナプスからのシグナルだと思うんですが、このように Z 軸方向にかなりシグナルが伸びていることがわかります。一方、3D ステッド顕微鏡で観察しますと、ここが同じ位置に当たるんですが、Z 軸方向へのシグナルの広がりは全くありません。また、この XZ 平面からも明らかなように、一つ一つのシナプスをほぼ完璧に分離することが可能になっています。さらに、個々のシナプスの形もかなりはっきりと捉えることができております。こちらのムービーにありますように、一つ一つのシナプスが大脳皮質でどのように分布しているか、そしてシナプス間の距離に関してもかなり正確な情報が得られる、そういう顕微鏡です。私たちすでにこの顕微鏡を人の脳組織にも使っております。この実験では、人の脳組織で立体的に抑制性シナプスを可視化するという実験を行っています。抑制性シナプスのマーカーとしてジェフリンに対する抗体で染色しました。先ほどの繰り返しになりますけれども、Z 軸方向の解像度が劇的に改善しているということが分かっていただけると思います。さらに 3D ステッド顕微鏡を使うことによって、一つ一つのシナプスからのシグナルの強弱、これがかなり客観的に捉えることが可能になっています。 最後の、例としまして紹介するのは、海馬 CA1 領域の神経細胞に脂質収縮型ビーナスを発現させて、抗 GFP 抗体で染色した結果です。こちらのムービーにありましたように、一つ一つのスパインのかなり細かい構造まで表面がくぼんでいる様子、そしてネックの正確な細さ、長さまでこの顕微鏡で定量することが可能になります。このように 3D ステッド顕微鏡は脳組織内でシナプスとスパインの微細形態を観察するのに最適な顕微鏡だと私は思います。現在この顕微鏡を使いまして神経細胞がシナプス形成までどのように発達するのか、そして発達障害のモデルマウス、患者の脳を使って発達障害の病態を明らかにするという研究に取り組んでいます。最後に下地になりますが、この顕微鏡に関するセットアップ、そして維持に関する実験、これに関してアビリオイとアビリオインストゥメンツ、そしてカンタムデザインの皆さんには大変お世話になってきました。さらにマックス・プランク・インスティテュート・オブ・エクスペリメンタル・メディシンのカトリン・ギリス先生は、私が初めてステート顕微鏡を使うこのチャンスを与えてくださった先生です。そしてニールス・ブローデ先生は私のメンターの先生です。この顕微鏡を立ち上げる際に慶應義塾大学の野添望様には大変お世話になりました。今日紹介いたしましたデータの一部は、群馬大学大学院医学系研究科の小宮戸先生、高橋先生、花村先生、小金沢先生、そして吉田さんとの共同研究として行われました。以上になります。あともう一枚だけスライドを、出したいんですけれども、今現在私たちの研究室ではですね、助教を募集しています。世界に発信できる研究を進めたいという方、研究に対する情熱なら誰にも負けないという若手の方、私たちと一緒に研究しませんか？我こそはという方、川辺までどうぞご連絡ください。ご連絡お待ちしています。以上になります。
はい、川辺先生、ありがとうございました。ここまでの川辺先生からの発表の中でですね、ご質問等、ございましたら、ここで受け付けさせていただきます。皆様のですね、質問、ボックスの中にご質問いただきましたら、都度回答させていただきます。ただですね、今回のウェビナー最後に、私の方からご登録いただいた皆様のEメールアドレスにアンケートを送りさせていただきますので、そこにコメント等いただいても結構です。特に皆様ご質問ございませんでしょうか？なさそうですかね。はい。はい、それではですね、次のセッションですね、アッペリア・インスメン社の、マーティン・マスケットより、実際に装置を使ったご説明の方に進めてまいります。マジ、please go ahead.
So, thank you very much. Thank you very much for the very nice talk, Professor Kawabe. I really motivate you to also send applications to him because he's a great scientist, and I think he will have a great time in his lab. And now I thank you very much also for joining from my side. I'm Martin. I'm an application scientist in Göttingen, so at abberior Instruments. And I would first of all like to talk a bit about the techniques we have, and then I'll show you on the microscope actually what we can do. So let's start. We are a small or mid-sized company which is basically involved in super resolution. We are one of the leading companies in super resolution microscopes. We provide STED microscopes in different flavors. We provide min-flux microscopes. Today we will focus on our flagship FACILITY Line, which is a STED microscope with a lot of expert features that can be or is designed for facilities and for multi-user environments. So we focus on the FACILITY Line today. Why would you want to do super resolution? Professor Kawabe motivated that quite well, quite great. But here just a comparison. If you look at, for example, the synaptic cleft or synapses in general, what you would see in confocal or wide field is mostly this blur. But what we are actually interested in as scientists is we want to look at the substructures. And these substructures, the synaptic cleft or here the vesicles, they are quite small. They are all well below the diffraction limit. So to go below the diffraction limit and actually resolve them, we either need electron microscopy, but there you have a labeling issue, or you need super resolution microscopy like STED, STORM, PALM, or MinFlux. So if you want to go for these fine details, you can either go for STED with this talk, or if you want to go even further with smaller field of view, you go for the MinFlux microscope. So that basically allows you to answer many more questions regarding colocalization, organization, and spatial organization of proteins. Why would you do that? Well, with super resolution, in this case with STED, you can get better models. For example, this paper by Elisa de Est and Stefan Hell regarding the node of Ranvier, you can see this advanced information about protein lattices and basically protein structures that can only be resolved by STED and super resolution microscopy. And then you get much better insight on how, for example, the nodes, the para nodes, and so on work. Another key advantage of STED, also motivated already, you can do live cell imaging. Here, this great papers by Katrin Willig, the lab here in Göttingen, where they did chronic live cell STED imaging on the left-hand side. And here they could even do 3-color in vivo STED in mice. So just to motivate it a bit, super resolution is also well established by now. But most of the super resolution samples are rather flat. For example, these neurons, adenosine spectrin, also shown by Professor Kawabe, a very nice example of super resolution. But here the key point is usually you do super resolution in the flat part of the sample. Yeah, if you want to do, for example, do this 3D rendering here and you want to go deeper into a sample, or you want to do advanced 3D reconstructions, what you are facing is aberrations. So meaning you want to have, you can have issues with complex samples, and you can resolve these issues for confocal STED with the given techniques I would like to present you to basically improve your images, get better and crisper and sharper images. We have here 3 topics. First one would be tissue imaging with our solution additive optics. Then we have imaging of crowded areas, so areas where a lot of labeling is present, where we look at the matrix. And if there's still time left, we'll also look at live cell imaging and check this. So, for example, here we have a kidney. So this is glomerulus from David Unas-Yes, a scientist here in Cologne. And this is a 100 micrometer cleared tissue. And you see here, if you go deep into the tissue, so this is basically at the cover slip, this is 100 micrometer deep. And if you go into the tissue, you can do STED up to roughly 20 microns, very fine. 2D STED, 3D STED, no problem. So cryostat sections are okay. But if you want to go deeper, if you have thicker sections, or if you have complicated samples that might have more embedding here, for example, you see the signal degenerates a bit because we have aberrations. So how do we remove these aberrations? Well, we can use space technology. We can use the technology used for telescopes by astronomers. They have a technique called adaptive optics, so they can adapt the image beam, the image light path, and get better images. For example, here you have an uncorrected image from Uranus, and then here is an adaptive optics image. So this basically helps a lot in improving image quality. How is that done actually? Well, you have an input beam, meaning you basically have your laser beam. You can put it on an adaptive optical element. In our case, this is, for example, the deformable mirror, a mirror with many individual movable subunits. And this mirror can then be finely tuned, and you can tune and modify the beam and correct aberrations. For example, here on the right hand side, you see it, a flat wavefront. So this is basically what's uncorrected. And if you go uncorrect into the lens, you have this aberrated wavefront. So you have many focus points here. It's a very long focus point. And if you have this adaptive element, you can form basically the wave of the laser, and you can improve focus massively, therefore getting much better quality. So how does it look like in practical terms? Well, if you use adaptive optics, you can do 3D STED down to 100 micrometer in complex tissues. This is important for thick tissues, but also for tissues which have suboptimal embedding or other technical problems that can be partially compensated with adaptive optics. This not only works for STED. Here's a typical confocal image of a relatively thin section, but here the embedding was not optimal. And you see, if I use the adaptive optics module, I can now flip between these two, uncorrected, corrected. Also, the confocal signal profits significantly from this deformable mirror. That would be one option or one way to improve the imaging. A second issue that can arise are crowded areas. Crowded areas are an issue if you have, for example, this sample here we labeled phalloidin, or we used phalloidin to label actin in CACO2 cells. So these are special cells of the gut epithelial. And you see here, the confocal signal, you get your structures. If I now do STED, I get much nicer substructures. Yeah, you see I get a significant increase in resolution. But what is still present is this haze here. Yeah, and now this haze is coming from nearby structures above and below the focal plane. Yeah, roughly in a 600 nanometer range, so something that the pinhole cannot easily remove. And you can actually now remove this haze without touching the foreground. So I get rid of this part and I keep the brightness here. How is it possible? Well, we can use a concept from nature. These are so-called striped horseflies, and they're insects, and they have very good 3D vision. They can very nicely fly and evade structures in the air because they can nicely see what is foreground, what is background. And they have these compound or complex eyes with many subunits. And the idea is that every of these subunits basically sees the same, but from a different angle. Yeah, so a different point of view. And these different points of view of seeing the same thing allow you to separate foreground and background. This is actually also the way humans do 3D vision. You can actually try that yourself. You can extend your arm, look at your thumb, and then open one eye, close the other eye, like our head of development, Matthias Reuss, is doing here on the right-hand side. And what you actually will see is that the background stays the same, but the thumb moves. It looks like it's moving in relation to the background. And this two detector setup, your eyes, can already discriminate what is foreground, what is background. And that can basically be used to separate signal and for us to not always hit any chair when we pass by, because we can actually distinguish where it is positioned in the room due to 3D vision. So how does it look like in function? This is again the CACO cell sample. Again, these are microvilli labeled for actin. And if I now use the matrix detector, I can significantly reduce the haze while keeping all the good sample quality. Going here more into detail, you can clearly see, especially these areas here between the filaments, you could not easily remove. Otherwise, if you wanted to do some image processing, that would be always artifact prone. Here we just measure the foreground and background and selectively remove the background. This is also very advantageous for 3D STED. Up here you see a nuclear stain. In this case, you see with 3D STED quite a lot of haze because the sample was not optimal, and you see a lot of aberration. And if you then use the matrix, I can really get rid of all this haze here and improve your 3D renderings. This looks like this, for example. You have your 3D rendering here. This is 3D STED. You already see there is some structure within, yeah, but it's covered by haze. And if I now basically do this matrix post-processing or the matrix detection, we can basically get all these structures much better and get better 3D reconstruction. So you get better models. This not only works for these CACO cells, but also for a wide variety of samples, and it has key advantages. For example, here, a neuron with allosin spectrin, on the right-hand side, Homer and bassoon in tissue. And they both suffer from these dense areas. For confocal as well as STED imaging, this can be tricky. And if we do matrix imaging for confocal and matrix STED, we can get an optical decrowding. Yeah, so we remove the haze, we improve optical sectioning, and this makes it much easier to get great images in tissue sections or in crowded cell samples. So before, super resolution was always done here in these outer parts because it's easy. With matrix, you can also do some nice super resolution here in the very crowded and overlapping parts with a lot of labeling. Again, here the synapse Homer bassoon spectrin labeled, and you can basically see how you can decrowd that better than just looking at confocal. Another example, myself, my background is in myelin biology, so I did my PhD in the same institute as Professor Kawabe at the MPI for experimental medicine, or where he worked, with Klaus Aminavo. They were interested quite a lot in myelin, and here you see MBP, so myelin basic protein labeled. And on the left-hand side, the normal confocal image. On the right-hand side, the matrix STED image. And you can clearly see you get much better clarity. You can really see the hollow tubes of the myelin now with matrix STED. So better resolution and better signal to noise, signal to background. And I would also like to briefly touch the topic of live cell imaging, as already presented by Professor Kawabe, but here is another couple of examples which you can nicely do with STED. For example, this movie here by the Stefan Jakobs lab in Göttingen, where they labeled live mitochondria, or here one of our own movies now. This is with a dye from the Peking University, an interesting dye for cells with large mitochondria, where you can nicely see the cristae using STED, and you can do nice movies. So it's a stable dye, and this can be basically then improved by using adaptive illumination, what Project Kawabe mentioned at least. So basically you minimize bleaching by really pointing the STED beam only where it's needed. Yeah, minimize bleaching, minimize cellular stress, and this can help improve live cell imaging because you stress the cell less, and it can also improve resolution, because imagine you have this image up here, the lamine and the nuclear pore complex, and these structures are very close by, and you know the structure is hidden here in the center, and I don't need to illuminate this part or the upper and lower part of the structure here with STED if I know by rescue and diamond that there is no underlying structure in these outer parts. I can really focus, and I can use all my available photon budget to really resolve what's important. So if you have a strong STED laser and you want highest resolution, adaptive illumination can help you in getting there. Now I would switch to the software. If you have any questions, feel free to ask them. Feel free to drop them in the chat, or also feel free to contact QD for further questions regarding abberior. Now I'll switch to the microscope. You see here I have my software in the back. This is the so-called light box. The microscope has a software designed to be user-friendly and be used by everyone. I went to, in one of the first weeks when I started with the company a couple of years ago, actually 2 years ago now, I went to Berlin and I trained 20 people using this microscope in actually 1.5 days in 2-hour slots. So it's very intuitive and straightforward. Important function here already, you have a full auto alignment, meaning you can put on an auto alignment sample, the microscope will detect the sample, will record from the sample, will adjust all its mirrors, because these are motorized, will adjust the pinhole and so on, and will do an alignment by itself. This takes roughly 3 to 5 minutes, and it guarantees you that you always have the perfect condition of the microscope, even if you haven't used it for a while. So now we go here to the welcome screen and I press new sample. Here are now dye chips. So for example, abberior star red. This is a dye. This is where it's exciting, or where it's emitting. So that's the color basically. And then you have here the options to either select a dye, inspect a dye, or clone and customize a dye if you want to adjust the spectra and want to set it up yourself. Let's inspect the dye. We see that there's an absorption spectrum, so where it is excited, and a fluorescence spectrum, so where it is detected, and also a deactivation spectrum, which STED laser to use. So in this case, the 775 nanometer STED laser is used. This information is then relayed to the microscope, and the microscope will automatically select the lasers. It will automatically select the detection window. Of course, you can change that, but it will automatically select the best choice. Now, I can drag this over here and select this dye. It's not only possible to show these recent dyes, you also have options to show dyes matching a text search. So in case you want to have, for example, certain Alexa dyes or certain abberior dyes, you can easily find them, or of course also DAPI and consorting. You can also go show dyes matching a specific emission wavelength, where I can basically play along and look for the dye based on its emission wavelength. I can also use dyes which are similar and modify them to get my image that I desire. There are already questions in the plane. That's good. We will answer them in a later moment. So I will quickly actually shut off my camera. So the screen you see is a little bit larger and now should be full screen. I click New Overview. And what I set up here is basically a confocal overview that I can easily take, that the microscope will do a spiral scan and will charter or will map the environment of the sample. I press open, and then we start with the following scan. So here, this is a neuron sample, in this case also adenosine and spectrin. First, we'll have just a look at the adenosine, and I'll do a quick scan. So here it will start in the center, and then it will basically spiral now around to roughly 400 by 400 microns, and I can look for a nice region to image. I can show that as well. If I do an overview, I can actually completely remote control this microscope. So while it is possible to use the microscope itself on the microscope controls, I can also do everything by mouse and drag and drop. So let me finish this. And then I press briefly New Overview. And you see now I can scroll with the mouse wheel, and this makes the microscope change the focus. Yeah. There's also a Z slider here, so I can also control it here if I like. But usually I do it by the mouse wheel. And I can also click and drag. So I want to center this part, for example, I click and drag, and then the microscope stage will move independently, and I can center a structure. That can be very helpful if you're looking, especially if you're working remote for whatever reason. I often do my remote sessions from home. So if I start my measurement and I want to check on it in the afternoon, or actually before going to bed, I can remotely log in and then modify the microscope to my needs, or select new regions, for example. So let's do a quick STED image here. I look for a region that might be interesting. Maybe we'll try this one here. So this would be one axon, I assume, or at least a neuronal process. And with the given part here, I can set this up. I will just do it now and explain in a minute. And what I do get is this very much improved spectrin labeling. So what I can really see is here, if I now overlay them, for example, I will do this here in magenta and this here in green. We can nicely see we very much improved our resolution by using STED. And the interesting thing is I can just select a region here, and then I just switch on STED and get a nice result more or less immediately. So that's super easy and straightforward to get your results. Everyone who can do confocal imaging can for sure also do STED imaging. So we have our one color STED. I would like to briefly explain what to see here on the left hand side. We see here the different ribbons. We have here the light path. The light path contains the objective and the pinhole. You can set that, but otherwise it's set automatically. And there is always the option to escalate to the next level, which would be this here. Here you can also adjust gating if you want, or the spectral range in which it is detected. For example, here 650 nanometer to 755 is a detection window for star red in this sample. The scan dialog is similar. It's very easy. You have here your excitation that you can set, and then you have here the resolution slider, which if you increase will adjust the STED power. Here you can adjust the image speed, so basically the dwell time per second. And here in the second escalation, you get more options. For example, adjusting the accumulations. An important part here, we are using avalanche photodiodes or APDs. These are virtually background free, or very, very little background. So we don't do any mean of the signal, but we do an accumulation. So we collect more and more signal, and we get very good results with that. So there is no averaging needed. Pixel size can be set here and adjusted to your needs. There's also the 2D and 3D slider, which I will come to in a minute. So you can do 2D STED, 3D STED, and you couldn't do anything in between. You can actually form your point spread function that Professor Kawabe mentioned before as you like, and put more power into the 2D donut or more power into the 3D donut. Let's maybe have a look here at the cell. I will do now an XZ scan, so I can basically section through the cell. I will do this with 40 nanometer, and we'll have a look here first. I will adjust it a bit, but now we are actually looking at the cell. Here is the cover slip. Here is the head of the cell, or the nucleus basically, or something similar. Now I will activate here STED for now, and we'll say, for example, 15%. And first we'll do a 2D STED image. This looks like this. And we see we get these thin needles, and we get significantly improved resolution in the X and Y, but at the moment not in Z. Yeah, so we get these needles, and this looks very nice in cross section. I can actually show that briefly here. So in the XY plane, it looks like this. We see the individual dots. And in the XZ plane, we see these needles. So you get improvement in 2D, but not so much in 3D. But we can change that. I'll pull the slider here to 3D. We do this again. And I press scan. And what happens now is that I get this different donut. The STED donut is now a 3D STED donut, and you see quite massively an improvement in signal. But what you actually also see here is that you have a lot of haze in this very standard sample. Here in the filaments, this is one of these axonal processes that doesn't disturb you so much because everything is flat. But here, with so many structures nearby, actually it might be a little bit problematic because you might be prone to this haze. And if you now switch on the matrix detector, I can do this here. For that, I will also briefly open the pinhole because the matrix detector works as its own pinhole. And I will press scan. And we see how good this nucleus looks like. Now the matrix will align itself to the specific wavelength, in this case to star red and the red detection window, and we can have a look. So, well, this is our nucleus, or our core of the cell. For now, it looks very much similar to the APD. And now if we do a matrix post-processing, we can significantly remove here this haze. So this is a live example. I didn't know how this nucleus looked before. I could not select it, so I just tried. And you see here, I can do nice matrix imaging. So what have I done? Basically, I can acquire an image with the matrix detector, and then I have here the option to do some post-processing, I either can do a matrix pinhole, means it will simulate a pinhole size from very closed pinhole to very open pinhole. This can be very useful if you don't know, or if you want to maximize the light collection. Then you always record with an open pinhole, and with the matrix, you can then later on adjust the pinhole to your need. This is sometimes also, I learned, important for electrophysiology. If you want to do some combined electrophysiology and want to see the whole synapse, you might want to open up the pinhole, and then you can do the recording of the matrix and decide for a better signaling pinhole later on. Same here, I can go for the pinhole. I can show you the comparison. This would be a completely open pinhole. Here, everything is fine. Here we are struggling a bit. And now if I do the matrix post-processing here, I can select different regions. For example, I can select here the differential detection. The differential detection gives me the amount of background to remove while the foreground is not touched. So let's do this. Let's put this to, for example, 2. And you can see here, much better signal, much clearer in this part here. Also, this area here with the filaments is better, but it's striking here in the nucleus or in the soma of the cell. I can also do this here, sharpening. This is a deconvolution algorithm, and this deconvolution works on the differentially detected image. So it works on this one, which means it has better raw data, yeah, because it can, you remove the background, you do deconvolution, that makes the deconvolution better. If I deconvolve this image, it's possible, but it doesn't look nice, because here, of course, you can't do so nice deconvolution. But here with that, I can, for example, set the iteration of whatever 6, and I get, especially for point structures, I get very nice deconvolution here. So I would also like to show you that the matrix can be very helpful on other occasions. You see here, I have a sample with several colors. Well, I create a new sample, and now I also include the adenosine. I can name the sample here neurons. I can call this adenosine and this spectrin. And I can also define an overlay color. So if I want to overlay them, how they look like, I want to have them in magenta and green. I go again, press live here. We can have a look. We, for example, now here I have this structure. I overlay them, then they get the appropriate colors, in this case green and magenta, and now I will basically look for a region which has appropriate signal here. I can move my stage now if I don't want to do an overview. I increase here, does this a bit. Maybe I have a look into this structure here. I can now define a region of interest. Yeah, this pops up here in the tree view. This is the overall overview, and then I have the sub image here. I can turn this and adjust it accordingly, and then I can try to get my image. So first of all, I will focus a bit. Here I press live and look for the right focal position with the mouse wheel. I'll just save this now, and I can now switch on STED. Let's say 25% of the STED laser here, and here, because the labeling is a little bit fainter, I will go for a little bit more power. This is always the excitation power. This is the STED power, but that's something we can discuss later in detail if needed. And then I can basically press scan, and I get my images. And what I can nicely see is, okay, I get my adenosine, my spectrin. If I overlay them now and adjust the contrast a bit, actually I make it a little less bright, we can nicely see this intersparse labeling that's fitting right in here. So that is how you can do easy 2-color STED. Yeah. It's also, if needed, you can also get a third STED color. Then you have the 595 STED laser. For time reasons, I will not use that, but in the end, it's the same. You then get a third color option here. You click the laser, and you can do 3-color STED one after another. Yeah. Not only does it work nicely for these two colors here, if you don't want a second STED laser, or if you're interested in, for example, the nucleus and you labeled DAPI, you can also use the matrix detector for that. I press here, I insert DAPI and star green. In this case, these are also neurons, but here we see phalloidin, actin, and here we see, as mentioned, DAPI. And now I can basically also here create an image. And we can see the deconvolution on the matrix as well. So we have here this part. I'll move it a little bit. I increase DAPI signal a bit. And maybe I'll find a nice cell. For example, here, I go to the center part. I can here increase the pixel size, so scanning is a little bit faster in a moment. For example, here would be a cell with a nucleus. And now what I can do is I can, for example, select here a region. Let's put this. And first of all, I will do a matrix scan with the green. So I'm doing confocal imaging with the matrix. It will now briefly align itself to the new wavelengths, in this case, to the green detection window. And once this is done automatically in the background. It will basically record a confocal matrix image of that region. Takes one moment. It's preparing the alignment. And now I get my image and I can do some interesting deconvolution. So first, I'll do some increased power, increased excitation here. And I say, okay, give me 10 accumulations to get a nice image for deconvolution with 50 nanometer pixel. And now this is a pure confocal image. This is how you get it out of the system normally. Yeah. And now if I copy this, press Control and copy a channel and do some post-processing here. Yeah. I can actually already see, okay, here for confocal, this also works. Yeah, I can get rid of this background. So every confocal image can be improved if you have this background haze issue. And now I can also do this sharpening here. And what you can see, I hope you can see it, is that you get a much crisper, much sharper image. And this all with confocal already. So the matrix detector gives you the flexibility to do advanced imaging with all wavelength modalities, so green to or blue to far red. You can do STED with it, you can do confocal with it, and you can use it, for example, for advanced deconvolution. Another nice example, which I usually very much like, are the nuclei, because here the DAPI signal, it's not steadable, and it's something you always have in your sample, and you might want to have a look at more specifically. So we do this now. I adjust again, and I set this to now the blue wavelengths. The system automatically sets this to the DAPI channel, and I will do some matrix imaging here. And without overdoing it, I will go here to a lower value because the matrix is very sensitive in the blue. I'll do, for example, 10 accumulations. I press start. You see we get a nice signal already here from the nucleus. And if I now do my differential detection paired with a little bit of sharpening, I can get much better substructures here. Again, we can compare it. If I open it to the maximum pinhole, what you can see here, you can get clearly better contrast and better ideas about your structure. This is also very nice for dividing cells if you look at chromosomes, for example. Without touching it too much, there are also these options for adaptive illumination you can see here, which can be used for this improved imaging. There's also options for adaptive optics on this system. If I had now a thick sample, I could now correct the spherical aberrations by pushing here around a little bit 2 sliders and get an improved imaging. But with that, I'm now finally finished for this part of the presentation, and I hope that you have some questions which we might be able to answer, hopefully. Thank you for your attention, and I'm looking forward to your questions. So I will go to the beginner slide to have a nice image. Do you have any questions so far? Would you like to see more about some special technique, some special effect? What would you like to see? There's a question. I hope I can. Shinya already answered some questions. Very good. I'm early, as you realized, Shinya. But yeah, please feel free also to translate questions for me if there's any need. Otherwise, I'm always happy to answer questions in general.
ご参加者の皆様、ご質問等ございますでしょうか？
I can also show more on the microscope if needed, but I'm done with the basic part. For the rest, we can of course discuss. Well, this is not the case. While we wait for maybe more questions, I can maybe show some more small features. For example, if I go to a new sample and we can again go for star red and orange, we can neurons. We can try again on a sample, and I can do a quick 3D stack. Might also be useful. I adjust here the settings. And for example, here, show you a matrix image on this. Maybe we first do it like this. I set here a little bit more power. As I said, this one is relatively faint. And here, and actually what I can also show you, that's something I really like, which I maybe do instead. I'll show you the Kako cells, because that's also a very nice sample. And so something you might have not seen before. And we can nicely demonstrate different effects here. So instead I will, if there's more time, I will just go to the CO cells and go for phalloidin, new overview. I'll select a region, and you see here, these are cells now grown on a filter paper. And we have here some structures. And for example, I can take one of these small isolates here, and I can go first for normal confocal signaling here. You see not much now. And if we now add the STED, so I use here the open pinhole, I add the STED laser, and I would go to maybe 15%, and here I go to shorter dwell time and number of images, and maybe 25 nanometer pixel or 30. It will now align itself again to the red because I changed the structures again. We wait for the alignment procedure, and then we can basically do a matrix imaging on this. So we got the question, are there are there any possibility matrix haze reduction reducing the signal? So the idea, if I understand the question correctly, so on the left-hand side, the confocal image, on the right-hand side, the matrix image. And if I do now some post-processing, it gets even clearer here. You see, you get much better reduction. And the idea is really, as the signal or as the system can differentiate between foreground and background, it can selectively remove this background here without touching the foreground. So this signal here is untouched, yeah, but this signal is diminished. But we can nicely see here that we can get very good resolution. Let's try also here. I can also copy settings from one image to the other. I did this by dragging image 1 to image 2, so that's also a way of intuitively selecting your settings. And you can nicely see here also these are the filaments you've seen before. And we can then do some post-processing. The post-processing is a linear process, so it's very well usable. And you see here, I can very nicely see and get my better resolution and my better haze reduction compared to, for example, when I open the pinhole completely or compared to the confocal. So I get significantly improved image quality. You can also try here a 3D STED. We go for 40, 40, 40. And first with the matrix, the pinhole. First we do a confocal scan. You see here that's the structure of the microscope of the sample. Go here for 15 and we can do a 3D scan. I do a couple of more accumulations here. And now I'm doing an XZ scan again. So we're scanning from the upper part to the lower part. Let's see how that looks like. Actually, it's very faint. So maybe this is not the perfect part for this structure, but in the end, what you can still see that you get nice recovery of the matrix effect through the microvilli up here. Any more questions I could answer? Could you show some time-lapse STED imaging with live samples? Well, yes, I could. I don't have any living samples with me. We could do that, of course, in a demo if you want, but that, of course, requires specific labeling. I haven't prepared any samples, but if you want to do, for example, live cell imaging, what you could do is you could select a couple of regions in the structure. Yeah, I can then tell every of these regions. I can say, okay, I want an interval of whatever 10 frames. Yeah. And of course more in or iterations and an interval of, let's say, 2 seconds. I press scan, and then what happens is basically that I get some live repetitive imaging here easily. So this now is doing, for example, 10 iterations and then goes to the next image, but I can also program it that I get one image, one frame here, then one frame for image 5 and one frame for image 6. So as I like, and I can then do this live cell imaging of samples. If you want to do live cell imaging for STED, I always highly recommend using snap tagged proteins. These with CRISPR-Cas, these are relatively easy to make, and with the snap tag proteins, you can use organic dyes, for example, our abberior live dyes, but also other dyes, for example, silicon rhodamine with a snap binding element that can help tremendously because these are very, very stable and you can do very nice STED imaging. But of course, you can also use stuff like this PKU antibody. So time-lapse imaging is possible. I don't have living cells right now, right here, but it's absolutely possible, and bleaching can be reduced with adaptive illumination. That's absolutely an option. We can also have a look at how to do a 3D stack here, just for out of curiosity. I could also show you how the software handles at the moment 3D. So for example, here I can go for live, and then I can select an upper plane let's go for this one. I can go here to this set slider and I can link it. I can scroll down, I can link it again, and I can here set the step size. Let's say we put it to 50 and 50, and then we can press scan, and we get a nice stack. We can also do that for 3D if needed, and you can basically get some nice imaging. And I can scroll through the stack now and project it. Another interesting thing I haven't yet shown is if you go here, you can either export it as TIFF, you can export it as OBF. The OBF is our raw file, which contains all the metadata and can directly be opened in ImageJ via the Bioformats plugin. As I said, you can open it in ImageJ. You can also open it in Huygens and do some deconvolution if you like. So there's a Huygens bridge and an ImageJ bridge for visualization. So with that, I think I would be done for this part. And I hope you enjoyed it, and I'm looking forward to maybe some more questions if you have them. Otherwise, I will switch now to the presentation. Thank you very much. Ayumi wants to share, would you like to share something? Yes, very good. The upcoming online symposia. Ayumi, we can't hear you yet. 1 second. Oh, yes.
すみません。ご清聴ありがとうございました。私、日本カンタムデザインの石原と申します。本日の内容以上になるんですけれども、最後に私の方から、今後のイベントのご紹介です。今回の、ウェブセミナーは、弊社日本カンタムデザインとアベリアインストゥルメンチャの共催のオンラインセミナーシリーズの中の第1回目になります。で、次回が、ちょうど1週間後の10月28日の木曜日、同じ時間4時からを予定しておりまして、内容が超解像顕微鏡の新しい手法であるミンフラックスというモデルのご紹介になります。でこちらが最大分解能2ナノメートルという非常に高分解能で画像取得できる装置となっております。でこの回では、ステファン・ヘル教授に最初にショートイントロダクションをいただき、その後、本日と同じように、アビリインストゥルメントのアプリケーションサイエンティストによるプレゼンテーションおよびライブデモンストレーションを予定しております。で、3回目が、1週空いて11月4日の木曜日、同じく4時から。で、こちらが、これから超解像顕微鏡を始めたいという方向けに、エントリーモデルのコンパクト顕微鏡ステリコンをフィーチャーした内容になっております。でこちらは先ほどのミンフラックスの方は英語でのセッションになりますけれども、3回目のステリコンに関しては日本語での講演になります。で内容としまして、プレゼンテーションおよびアプリケーション紹介、また、弊社のラボの方に実機のデモ機がございますので、同じようにライブデモを予定しております。で登録は弊社のウェブサイト、もしくは、この画面に表示されている QR コードの方からご登録いただければと思います。でもし、当日都合がつかない場合でも登録いただいておけば、後日録画の視聴が可能ですので、ぜひともご応募いただければと思います。で最後に、先ほど最初に花村の方からご説明がありましたけれども、終了後にアンケートをメールで送付させていただきますので、今後の参考までにご回答いただければ幸いです。本日は長時間ご清聴ありがとうございました。今後ともよろしくお願いいたします。
Thank you also from my side, and I hope you join for the other webinars. If you have any questions, just get in touch, either via QD or other means. Feel free, and I hope you have a nice evening. Thank you very much.
ありがとうございました。